Hey, hello everyone. For our second talk today, we will welcome Hannah Scanlon from Duke University, who'll be talking to us about microtubules. Thanks for being here.
Great, thank you, and thank you for the invite to talk. I'm excited to talk to you all about my PhD research, which is another math bio related topic. I've been looking at mechanisms of microtubule polarity, which support neuronal function and regeneration. So this work has been done in close collaboration with a biology lab at Penn State. And the image you see here is an image of a fruit fly larva, and the primary sensory neurons of the larva have been fluoresced. And those neurons are where we focus our research, we focus our interest, and get a lot of clinical measurements and clinical data to try to understand the behavior of microtubules, and especially the behavior of microtubules as they relate to neuronal regeneration. So I'll give a little background as to the biological context here to start before getting into the mathematical model. First, what are microtubules? Microtubules are polarized filaments that have a distinct plus and a minus end. They provide structure to the cell, and they also facilitate cargo transport. So there are different types of motor proteins that will walk along a microtubule towards either end of the microtubule while dragging cargo along with it. So in this way, microtubules facilitate this cargo transport. Specifically, they can facilitate really long-range cargo transport. Another key feature of microtubules is that they are dynamic filaments. They're constantly going through growing and shrinking behavior. So while the general structure of these microtubules are stable in order to support this transport and the structure of the cells, they're constantly undergoing shrinking, growth and shrinking behaviors that lead to pretty frequent turnover of the microtubule content, where the lifetime of a microtubule is really only a couple minutes. Okay, but why do we care about microtubules? Well, broadly, our work is motivated by neuronal regeneration. So to understand that, I want to first orient you to a neuron. There are three primary components of interest in a neuron. The first is the cell body. The second are the dendrites, which is this branch system that spreads out around the neuron, around the cell body to receive signals from other neurons. And the last is the axon, which is this long, thin branch which sends signals from this neuron to other neurons. Now, in your body, you have two different types of neurons. You have neurons that are part of your central nervous system, which is all the neurons in your brain and your spinal cord. And you have neurons that are part of your peripheral nervous system, which is everything else. Broadly speaking, the nerves in your central nervous system are unable to regenerate after traumatic injury. That's where you end up with things like spinal cord injury, where the neurons are not able to regenerate. However, neurons in your peripheral nervous system are able to regenerate. So if an axon of a neuron is severed, that axon is able to be regrown. And so it's really motivating to try to understand how these peripheral neurons are able to regenerate their injured axons to potentially identify what is going wrong in the central nervous system. This behavior is also pretty well conserved across species, and so we see similar behavior happen in the Drosophila larva that we are interested in, where we see those neurons being able to regenerate. Now, to tie this together, how are microtubules related to this neuronal regeneration? Well, to understand that, you have to understand how an axon regenerates. So here we have another picture of a neuron. And what happens is if you sever the axon of a neuron, one of the dendrites will, in some cases, be converted into a new axon. And one of the primary behaviors that has to happen for a dendrite to convert into a new axon is for all of the microtubules in that dendrite to reverse their polarity. In particular, in most dendrites in cells, most of the microtubules are facing minus end out, so with their minus end facing away from the cell body. And in most axons, microtubules are facing plus end out, with their plus ends away from the cell body. So for a dendrite to convert into an axon, all of those microtubules need to have their polarity reversed so that it can grow as an axon. Our research really tries to understand how these microtubules work inside of these systems, what regulates the behavior that allows them to maintain this really healthy, stable array over the lifetime of a neuron, and tries to get at what we might be seeing, what behaviors might characterize this reversal behavior after axonomy. A little more detail as to kind of what we're looking for in this model. Microtubules play a couple key roles in neurons that we want to capture. The first is that microtubules, like I said, they facilitate long-range intracellular transport with a stable polarized array. So the microtubules in the axon of your cell are all facing in one direction. The axons in the dendrite of your cell are all facing in the other direction. The key component is that the microtubules in different regions of the cells are all aligned in the same direction. And this is stable over the lifetime of the cell. So the cell is able to maintain this polarized array over the lifetime of the cell, which in our Drosophila larva, for example, that's about 5 days. And they do this despite the fact that microtubules are dynamic and they're turning over on the timescale of minutes. The last thing that behavior we want to capture of these microtubules is the fact that they can rearrange in response to axonal injury, especially in these peripheral neurons. So that means we want to try to identify mechanisms that are able to accomplish those first two goals without being locked into one particular polarity array, because we know that that's not what is true and that's not what happens in these neurons, as we see evidenced by the behavior after injury. Okay, so let's boil that down to slightly simpler terms. What does that mean? We want to identify mechanisms that do. First, we want mechanisms that produce a polarized microtubule array. We want that array to be stable over the lifetime of a cell. And we want it to be potentially adaptive to injury with some injury mechanism. So my work tries to use a previously developed model of microtubule dynamics, apply some mechanisms that have been identified biologically, to really get at these characteristics of microtubules that we see in cells and try to identify what mechanisms characterize these behaviors. So to start, I'll introduce you, this is probably the most mathematical piece that you'll see in this talk. Start with the model that we use for microtubule dynamics. That model is a stochastic model. It's a Markov chain model that represents either end of the microtubule as being a Markov chain that is either in a growth or a shrinking state. The switching rates between growth and shrinking are characterized by a time component. And then while either end is in growth or shrinking, there are different growth and shrinking velocities that dictate how much that microtubule grows or shrinks. There are a couple of length limiting mechanisms that some of my collaborators developed while developing this model, including a length-dependent catastrophe mechanism, where there's some evidence to show that the length of a microtubule, the longer a microtubule becomes, the more likely it is to switch to shrinking, as well as a mechanism that regulates how much of this growing material you have, which is called tubulin. But all that said, we put that together and we end up with this really realistic model of what microtubule dynamics look like. So here you're seeing a simulation of 10 example microtubules where, again, this Markov chain is dictating their growing and shrinking behavior. You see them relatively quickly dying, and then we are renucleating them. So we see these range of different lengths of microtubules, these different behaviors. They move a little bit. Overall, a lot of the behaviors that we see happening in our living systems, we see captured pretty well by this model. So my work looks to take this model and take it a step farther and really get to that spatial context that I introduced you to earlier. In particular, we look at a spatial context which represents what we call a comb dendrite. So this is where a lot of our experimental research focuses. This comb dendrite is seen in a lot of neurons in our Drosophila larva. And the nice thing about this comb dendrite is that it's really easy to identify where new microtubules are being nucleated, as almost all of those happen at what we call the branch point. So the point where the main trunk of the dendrite meets these side branches. So we're going to represent the same geometry with a slightly simpler version, where we're just going to represent that main trunk of the dendrite. We're going to represent those branch points as locations where microtubules will be nucleated. And then we're going to run simulations of these microtubules, where you have this vertical domain. Microtubules are going to be seeded with random polarity. And then as the microtubules grow and shrink, they will die, and we're going to explore mechanisms which regulate the polarity of this microtubule nucleations. There are a couple of mechanisms that have been identified by our collaborators that they believe to regulate which direction a microtubule will grow after it's nucleated. So the goal of this project is to explore those mechanisms, implement them in this model, and see if those mechanisms are sufficient to produce that overall behavior that we're looking for. The first mechanism that was identified by our collaborators is what's called a nucleation feedback mechanism. Experimentally, the data that they had basically said that the polarity of new microtubule nucleations matches existing polarity distributions. So what this means is that in an environment where the majority of microtubules were initially plus end out, the majority of new nucleations are going to match that distribution as mostly plus end out. However, in an environment where most microtubules had mixed polarity, we had about 50-50, then new nucleations will match that distribution as well. What that looks like in our model is a very simple implementation where, again, we're going to nucleate just at these specific nucleation locations. And the direction that that microtubule nucleates is going to be dictated by the fraction of microtubules that have that direction in that local content. The other mechanism that was identified by our collaborators is what's called a checkpoint mechanism. What this mechanism looked like experimentally is it told us that new microtubule nucleations have an increased probability of catastrophe that seems to be dependent on parallel pre-existing microtubules. So what this means is microtubule is nucleated, sometimes it will grow and it will just survive and continue growing. Sometimes it will catastrophe very quickly. And it seems like that catastrophe rate is dependent on there being other microtubules around it that have the same polarity distribution. There's a lot of reasons biologically to believe that microtubules do a better job growing or more stable when there's other microtubules around them with the same distribution. So we work to characterize this data in a mathematical context in order to design a model implementation for this mechanism. It took us a while to represent the exact probabilities of success based on the different environments and the different orientations of nucleation. We ultimately developed this mechanism that has two steps to it. The first is that for any new microtubule nucleation, there's an automatic probability that that microtubule is just going to die. It's not going to survive regardless of what's around it. The second step is that there's the survival of the microtubule is dependent on it seeing matching content, so it observing a microtubule around it that has the same polarity as it wants to grow. So identified with this mechanism development that there's actually probably more going on in this mechanism than our biologists originally believed. That there's this automatic chance of failure, and then there's this survival that's based on a subset of local microtubules. Okay, so put all that together, what are we doing? Well, we're simulating again this trunk domain of this dendrite. We are initializing our microtubules as half of them having minus and out polarity, half of them having plus and out polarity. We've implemented both of these mechanisms that we carefully designed to represent the biological context, and we want to see what behavior does this produce? What we see happening pretty quickly is that all of the microtubules in these simulations go to the same polarity distribution. So for example, in this middle simulation, we see all of the microtubules quickly reached a minus and out polarity. In this far right simulation, we see all of the microtubules reached a plus and out polarity. And the left, it's still kind of deciding which way it's going to go, but we're starting to see alignment of these microtubules. So what we're seeing broadly is that these mechanisms seem to be sufficient to establish this biased polarity distribution that we see happening throughout different regions of the neuron. In particular, if I look a little bit closer at some of the results from these simulations, we can look at what percentage of microtubules have minus and out polarity over time in two example simulations. So here we're seeing one simulation in blue where all of the microtubules quickly went to 100% minus and out polarity, and a simulation in red where they pretty quickly went to plus and out polarity. You see them leave those extremes occasionally, but they're staying pretty consistently up at those polarity distributions. If we repeat this for 100 different iterations of our simulation, we see that at the end of a 5-day simulation, that all of the microtubules end up either in this 0% minus and out polarity or 100% minus and out. So again, showing the consistency of our mechanisms at establishing this biased polarity. So what we've done so far, what we've noticed about these mechanisms is that we've identified unbiased mechanisms which regulate microtubule polarity at nucleation to establish a biased microtubule array throughout this linear domain we're interested in. Okay, but we weren't just interested in establishment. We were also interested in seeing maintenance. Again, we see this biased polarity distribution being maintained over the lifetime of our organism. So we want to look at how well our simulations are maintaining biased polarity. To do that, we plot a cumulative distribution function of the time simulations took to hit, depart, and return to a fully biased polarity distribution. And we see it takes all simulations reach a fully biased polarity distribution within 2 days. It takes as long as 5 days for simulations to ever depart that 100% minus and out or 0% minus and out polarity. So it's relatively stable on that time scale. And then even if they do depart that fully biased polarity distribution, we see that they return quite quickly often. So most return almost immediately, but all we see returning to that same biased polarity distribution within a day. So we can also say that our mechanisms now have demonstrated that they maintain a bias polarity over a timescale equivalent to the lifetime of an organism, given that the lifetime of these larvae we're interested in is really just the timescale of 5 days. Okay, so all that work is stuff that we just recently published, actually, to kind of talk about these mechanisms, understand how they contribute to polarity regulation in an organism. But what we really were interested in getting at was that adaptive injury response in neurons. So taking you back again, what do I mean by this injury response? Um, Again, we see what we call a polarity reversal in a dendrite. So if you sever an axon, what we see happening is that one dendrite will reverse all of their microtubules from a minus end out distribution to a plus end out distribution. That naturally raises the question of, well, how do they do that? So our experimentalists have done a number of experiments exploring and trying to identify what they see happening and what could possibly be mechanisms to produce this reversal. And one possible mechanism they have identified is potentially entrances of new microtubules from the cell body. So before injury, there's a very low success rate of new microtubules trying to enter a dendrite from the cell body. In particular, these are typically plus end out microtubules. So we're seeing that the dendrite, which is mostly minus end out, doesn't really want plus end out microtubules to come from the cell body. They have a very low percent chance of success. However, after injury, we see that success rate increase dramatically. So we see now in a region that used to be 100% minus end out, we're now seeing this influx of plus end out polarity from the cell body. So that raises the question of, is this just an artifact of some other mechanism that is producing this reversal? Or is this perhaps a mechanism that can produce this reversal in polarity in our system? So to understand that, we can test reversal. We can test what reversal does to our model, what a source of plus and out content at the cell body does to our model, and see if we get reversal. So we're simulating a very similar domain, an identical domain, the same mechanisms. The only change with our initialization of these simulations is that we're now initializing all of our microtubules to be minus end out, trying to represent that dendritic trunk. And then what we're going to do is we're going to introduce plus and out microtubules at the cell body. We're going to see if those plus and out microtubules are enough to cause a reversal of the polarity throughout this dendritic trunk. I simulated these simulations for a number of different nucleation rates for microtubules coming in from the cell body. And first we see if there's no microtubules coming in from the cell body, so a zero nucleation rate there, we see this minus and out polarity still pretty well maintained. So this is kind of the same results I just showed you earlier, that the system without any interruption does a pretty good job of maintaining bias polarity. However, as you start to introduce plus and out microtubules, even if you're introducing them at a really low rate, we see reversals start to happen pretty quickly in our system. We see that reversal is also dependent on the rate at which new plus and out nucleations are entering the dendrite. But we're starting to see that in this model, a proof of concept that microtubules entering from the cell body might really be that mechanism that's involved in this axon regeneration that we see happening in these injured neurons. So with that, some of the kind of key results we've talked about today. First, we developed a realistic model of microtubule dynamics in vivo. Then we designed a realistic polarity mechanisms, which recapitulate the local experimental data. So that was our nucleation feedback mechanism and our checkpoint mechanism. Then we demonstrated that establishment and maintenance of biased microtubule arrays on a timescale equivalent to the lifetime of the cell. And then finally, in this most recent results, we demonstrated the reversal of polarity can be mediated by introducing that anti-parallel content from the cell body. With that, I'd love to thank my collaborators, both my mathematics collaborators, Veronica, Scott, and Anna, as well as the biologists, Melissa and Gabarni, who have done all of the experimental data on this work. I'd be happy to take any questions.